Yeah. Okay. I'm recording now. Sorry about that.
No problem.
Okay. Let's go back here. So you see here on the right, this is a Volcano plot that is similar to the one before, but now there is an extra quantitative dimension in the graph. Basically, what you have again the fold change, you have the p-value, and then the size of the bubbles represent how abundant that protein is. In this case, was an HIV infection. And you see that, for example, here among the proteins that are not changing, proteins that are very, very abundant, there are these huge bubbles. These are probably ribosomes or histones. And then here you have, the proteins that are interesting. So now, if you scale your bubbles by protein abundance, this helps you to choose a priority target. You see that, for example, there is a protein here that is changing, but is extremely low abundance. And here there are proteins that are changing, but they are very abundant, right? So that actually is easier to point your eye at the things that are, changing and abundant. And actually, this was an HIV infection. What these are the HIV proteins. So that's not surprising, but it was a good positive control. So now it comes, after you displayed your data in a way that is helping you, to decipher, am I data, is there any change between my samples? Now you can start to include into your data representation biological properties, right? And the good thing about proteomics is that you don't really start from zero. There are databases that are rich in annotations for the proteins that are, in your list. For some organisms, of course, the annotations are much worse than for others. But for example, if you're working with, human proteins, they have a lot of information. And then, for example, let me 1 second mute everybody. Okay. For example, if you are working with, if you want to display, for example, show me the proteins involved in RNA and, where they are in the graph, then, you can easily, you can do it in Excel. You can basically overlay 2 Volcano plots in Excel, one where you show all the data, and then when you show only the proteins that have some kind of biological properties. And then you can see how they are distributed around the graph. So this is starting to give you some information about where your proteins of interest are. Are they regulated or not? And then actually, you're supposed to probably do this first. But let's say if you know nothing about your sample, one initial thing to do is to work on quality control. So how are my data, how are my replicates similar to each other? How are they different from between different groups? And there are many ways to do this. And to be honest, none of this is super informative, but it's a good exercise to make sure, for example, that your replicates are somehow similar to each other if you have global changes in protein abundances. So, for example, one way is to display them as a scatter plot. What you see here on the graph, this is a multi-scatter plot, and you can see, the more they align on a linear, on a line, the more the different samples are similar to each other, right? Because they have a higher correlation. You can express the correlation with a number, and you can see which ones they have a higher correlation or lower correlation. Or you can use one of these unsupervised clustering methods, like, for example, principal component analysis. Now, without going into much details, this type of data representation, they basically put close to each other samples that have an overall higher correlation by looking at multiple properties of the data. And you can use this, for example, to tell which proteins are the most which samples are the most similar to each other, and are there groups of samples that are more that affect more my proteome. For example, in this case, These are uninfected cells, and these are cells infected by adenovirus and herpes virus. And here there is what's the name of this one? Papulovirus, something like that. That's the one that affects your protein regulation the most. And also the distance between replicates, it also gives you a feeling about how biologically reproducible this type of analysis is. So this virus here, the way data are distributed is telling you that it's more challenging to have this virus to have always the same expression, always the same proteome regulation. And here I wrote down a little note. The proteomic tables are usually shorter than the RNA-seq table because of the sensitivity of mass spectrometry and because of the dynamic range of the proteome. So, expect that, don't expect the super tight correlations that you get, for example, with sequencing, but still, proteins should have, you see here from this number, they should have a correlation that is higher than 0.9, in terms of Pearson R-squared. So another property, which is a good quality control, is, how sensitive my experiment was, right? So even if you know nothing about, proteomics and mass spectrometry, you ask, for example, a service to analyze the proteome for you, you're going to get a table, and then you are going to be able, you need to be able to decide whether this is a reliable data set or not, right? And one of the things you can look at, for example, is how replicates of the same type correlate, right? That's the graph that we discussed before. Another one is how many proteins do I detect in my experiment X or my experiment Y? So this one seems like an easy check. In reality, the number of proteins you can detect from a sample, it changes a lot depending on the type of sample that you have. Even if you're working with human cells, there are some tissues and fluids where you can identify 4, 5, 6, 7,000 of proteins easily. For example, from a cancer cell, because cancer cells, they express a lot of proteins. But then, for example, if you take blood, in blood, it's harder to detect more than a few hundred proteins, because in blood, there are some proteins that are extremely abundant, like, for example, serum albumin. And then there are other proteins that are very low abundance. They get suppressed by the albumin. You're not going to be able to detect much more than a few hundred samples. So that's, how many proteins I detect in my experiment could be a very useful information to have, although you need to be a little bit familiar with the type, the amount of proteins that you expect to identify from whatever tissue you are looking at. And then, now let's say that we have plotted our data, and now we found that there is a number of proteins that are significantly regulated in our data set, right? So now, what do we do? How do we start exploring what are the properties of those proteins, even though we know nothing about them? And like I told you, there are all of these databases that they contain a lot of information that, in terms of annotations of different proteins. Even if you have an organism that is very poorly characterized, what you can do, you can blast the protein sequences of this organism, and then you can retrieve annotations from, let's say, human proteins that are the most similar to that protein. It's not perfect, of course, done this way, but it helps you to get a feeling in terms of bulks of regulated proteins. What are the most regulated functions? And then, what people do is they just take this regulated pool of proteins and then they perform gene ontology analysis, which is basically looking for functional enrichment. This gene ontology analysis is a good first step. It's very underwhelming in the sense it gives you a lot of information that are not very specific. So it's okay to do it, but at least do it the right way. And I'm going to tell you what the right way is. So the right way is when you look for the properties of your regulated proteins, these regulated proteins, they have some properties that they need to be compared with the background in order to tell how unique are the properties of the regulated proteins. Now, to compare this to the background, you need to have a background that is, appropriate, right? For example, I'm going to give you a quick example. If you are working with cells from muscle, right? And then you treat these muscle cells with a certain drug that activates, I don't know, some kinase receptor, right? So you're going to have some proteins that are being regulated by your stimulation, and then you input these proteins into one of these gene ontology tools, like for example, David. What David is going to tell you is that your regulated proteins are muscle-associated functions, right? You know that already. You're working with muscle cells. Of course, the proteins that are regulated are proteins that are probably specific for muscles. What you care about is what are the properties of proteins that are stimulated by my treatment. And the reason because you get as an enrichment muscle cells is because your background by default is the entire proteome annotation, right? So this is wrong because of course, whatever is regulated in your muscle cell proteins is going to be muscle related function. What you need to do, you need to use as background, you need to use, for example, the list of proteins that are not regulated in your cell because they are also muscle cells. And then the software can tell you what is specific in terms of proteins that are actually regulated. So there are different tools to do this. David is one, Gorilla is another. Actually, that's my favorite because it's the most intuitive and the quickest to use. The thing about Gorilla is you can have, when you open it, you have an input format that looks like this. And then what is really important is this part right here. So how do you input your gene list or your protein list? So you can either input it as a single rank list of genes or two unranked list of genes. And this is actually critical for what I just explained. Like if you choose the two unranked list of genes, it means that you will have a set of proteins that you want to find what is specific for them. Let's say your protein is regulated by the stimulation, and your background, what you use as your background, use all the proteins that are identified in your analysis. So all the muscle proteins, proteins that, coming up from your sample. And in terms of, and then what you get as an enrichment, you get proteins that are specific for your stimulation. If you instead use the single rank list of genes, you need to input your data as ranked. So ranked basically means you put on the top of the table the proteins for which you want to find out the specific annotation. And then, for example, you can take your Excel table and then you can sort them by, for example, most, best p-value. So the best p-value is on top, and the least significant p-value is on the bottom. You can just paste your list of proteins like this, and then it's going to tell you what is specific in the top of the table compared to the bottom of the table. So enrichment analysis is always a comparison, something to something. So always make sure that you take the proper background. Yes, and in terms of enrichment analysis, functional analysis, you don't have only gene ontology. Gene ontology is a database, and gene ontology database includes information like generic biological functions of your proteins, generic cellular localization, and generic, molecular process. So what is the protein involved in? Is it processing nucleotides, for example? And at the end of the analysis, it tells you something about, for example, what are the properties of your regulated proteins, it doesn't tell you much in terms of to which pathway they belong, or for example, are they specific for a specific disease? So for that, you can use other type of tools. Here I put just two examples. This is the KEG, which is the Kyoto Encyclopedia of Gene and Genomes, which looks specifically at pathways, because your proteins are grouped not in terms of properties, but in terms of to which pathway they are normally associated. And this one is a GCEA, which is another tool actually developed, I think, by the Broad Institute. And this one instead has a lot of information regarding which proteins are regulated in which disease. And then if you input your list of proteins that are significantly regulated in your analysis, it can tell you if this is known to be a specific protein properties of, you know, a certain type of abnormality for a cell. So again, think about what is your background, think about what kind of protein property you want to highlight, and there are different tools available online. And I think at the end of the presentation, I'm going to send you the slide. At the end of the presentation, I think there is a list of all of these databases that you can use and how to use the software. In the end, if you think about it, once you have your list of regulated proteins, and after you know something about what these proteins do, then you don't have to look anymore at data just like this. Now you can start to put them in context, right? So this graph that you see here on the right is nothing else that taking your proteins that are significantly enriched in the HSV. So here was the herpes infection. You know, the color code of the bubble tells you how enriched they are, and you can start grouping them by properties now that you have done a functional enrichment. So here is you start to actually look at proteins not anymore just in numbers, but also in terms of spatial distribution for those that have similar, you know, biological functions. And now it becomes much easier, you know, to recognize your data. Right? And then you probably notice that, you know, your proteins are connected by some lines here that are very faint, but you can see them in the background. So the next question is, how do I know that these two proteins have, you know, some sort of interaction of some type? And for example, I don't know, this one and this one, they do not. And here is, you can actually create these networks using other type of software that don't tell you necessarily, no, they tell you that too, but you know, their main job is not telling you what is the biological function of that protein, but they tell you how do we know that these proteins have, you know, some sort of genetic or physical interaction. Physical interaction means that they are touching each other in the cell somehow. Genetic interaction means that they're not necessarily physically touching, but one can affect the other, or they have some kind of very common properties of some sort. And for this, you know, we have other type of databases. String is probably the most famous software to do that because it's probably the easiest one to use. But you know, together with String, there is a list of many others. I think I have the list in a couple of slides. Intact, BioGrid, Bioplex. You know, there are other type of websites where you can look at whether proteins are known to interact with each other. So what you do here in a software like this, you have your pool of regulated proteins. Even better if you filter them for properties. So I have a pool of regulated proteins. I take all the proteins involved in RNA, and then I just paste them into this search engine tool. And then what happens is that you will get an output where proteins are linked to each other by stronger, you know, interactions. So these edges, these lines here, in this case, they are colored in a way that they are more dense. The color is more dark, is more dense. If there is a strong score in terms of protein interacting with each other, and that's how you connect list of proteins that are, you know, that are in how can I say, that are in your data set, they are regulated, and you want to know which ones they form, for example, specific complexes. and that's how you just paste your protein list, and then you get out a network like this. Be careful to not paste. Be careful to be conservative with the filters. If you paste too many proteins, then you get like a network of hundreds of proteins, which is hard to read. So that's what I say. Like, first look at what is significant, then look at what kind of properties you want to input, and then input that pool of proteins, and that's what you get in the end. In the string website, it also gives you some information about, for example, the functions of this protein. So if you click on any of these properties, then it actually colors your network, depending on what you click on. Although keep in mind that those enriched functions that are enriched compared to a background, where the background is the entire proteome, right? So it's not specific for your type of cell that you're working on. And then this is just a button to export. And then to make that nice graph, this one that you saw right here, there are some free tools online that you can play with the aesthetic. The most useful, to the best of my knowledge, is a software called Cytoscape. So once you export, these networks generated by string, if you import them in Cytoscape, you can play with the graphics. So you can color them the way you want them. You can move them around. Is a free tool, and is actually, I'm pretty sure that is the most widely utilized tools to display proteins as networks, or anything actually as a network. Right. So like I told you, you're going to have a list here of different databases where you can study protein-protein interactions. String is the one that is mostly used. There is a bunch of others. You see here that there are some notes about why they are different. For example, Bioplex is a database that is made by a lab at Harvard, and then that is exclusively based with mass spectrometry data. So the protein-protein interactions that are annotated in that website, they are the result of immunoprecipitation experiments of specific proteins. So you can see, which proteins interact with which from mass spec data. It could be a useful positive control if you are doing an immunoprecipitation yourself. Right. Now, let's say, for example, that you know nothing about how your proteins are interacting with each other. Let's say that you're working with a very strange organism, and you go on string, you paste your protein in that database, and no connector lines come up. So there's nothing known about how proteins interact with each other. How can you start grouping your regulated proteins in a way that it starts to make some sense about which proteins potentially can have a similar function? So some proteomic scientists, they've been thinking about that as well. So they say, okay, what if you know nothing about your proteins and you want to group them in terms of which ones are probably having similar functions? So what they did, they created a couple of websites. This is one called Co-Expression. There is another one called Proteome HD, for example. And these websites, what they contain, they don't contain any information about protein interaction, but they contain tons of data, like hundreds of experiments about protein regulations. So protein regulations basically means, this protein is abundant in sample 1, low abundant in sample 2, and so on for hundreds of samples. And then what you do, for example, if you have a set of, 50 proteins, and then you input them in the software, and then you want to know if some of them, they have some sort of shared property. What the software will do for you, it will group together proteins that are always found to have the same regulation across all the data sets that are in these databases. So for example, here you have, actually, let me start from here. Here you have, let's say, 2 proteins here, this one and this one. You know nothing about whether they interact or not, But when you input them in these databases, they are always found to have the same co-regulation. So when one is downregulated, the other one is as well. When one is upregulated, the other one is as well. And so on for hundreds of samples. This basically, what the software will tell you is that these proteins, they probably have similar properties because they always associate in expression. So this could be, this could be a useful type of information that you have when you have, when you don't know about any properties of your pool of regulated ones. Like for example, here in this graph, this is H.1 here in purple, and then these green lines are other proteins that you input that have the same relative abundance in all of these samples as H.1. And then you say, okay, I don't know what these green proteins do, but they probably do something associated to H.1 because they are always found with the same co-regulation. Okay, so everything we've discussed so far is related to protein regulation, right? So once you find your enriched proteins, now the next question is let's try now to find the biological meaning for these proteins. Now the thing becomes more complicated when you're looking at post-translational modifications, because a protein can have multiple post-translational modifications, and then you can have a protein where you have a modification becoming more abundant or becoming less abundant upon a stimulus. So now, what is the function of that post-translational modification? Because the function of the protein is not anymore informative enough if one modification goes up, one modification goes down. What is the function of the modification? It can become, really complicated very quickly because some proteins, like for example, histone proteins, they have like hundreds of possible modifications. That's actually what we do in our lab. We study histone modifications, and this is just a cartoon showing you, the type of modification that can be identified on histones, and here is how decorated histone sequences can be. Actually, this table is old. There are many more now. But this is very important because a protein can affect, a modification can affect the localization of the protein. Like there are some phosphorylations that they move enzymes from the nucleus to the cytoplasm. Modification can affect the lifespan, right, of a protein. One obvious example is ubiquitin, right? If a protein is ubiquitin-related, very likely will undergo degradation. Modification can affect interactions, of course. If two proteins are interacting with each other, if there is a modification in between, this could prevent the interaction. And then, of course, modification affects the activity of the protein, right? If, especially with phosphorylations, if a protein is phosphorylated, could be involved in some activity. If it's dephosphorylated, is not involved anymore. So it's the properties of post-translational modifications can be extremely critical and it can be extremely difficult, to identify because we don't have a lot of information about what is the function of each modification on a protein. So let me skip through this part. This is just a couple of slides that shows how do you enrich for post-translational modifications. We're going to get back to that in a moment. And here is just to give you a feeling about how you can represent, these modifications. Right? This is a network similar to the one that I showed you before. Also, this one is done with the Cytoscape. And then the question is now, how regulated are my modifications? Like in this study, they are specifically phosphorylation. It could be something else. If you zoom in, you see that you can have some proteins that have multiple phosphorylations, and then they can be differentially regulated in different samples, right? So for example, this is a specific phosphorylation that is down-regulated in the second sample, not regulated in the third sample, for example. And so, like I told you, like it becomes really tricky because it's not enough to know the function of the protein. You need to know what is the function of this phosphorylation compared to this one, for example. So how do we tell what is the function of a post-translational modification? Well, that can be really tricky, but there are some steps that you can proceed with that simplify your interpretation. One of them, you can study, for example, are my regulated modifications always in a specific protein motif, right? So your modification will be in a certain location, and then you're going to have some amino acid flanking your modification. And what is useful to know is that if these amino acids that flank your modification, they always have, the same, right? Because if they are always the same, what you can do, for example, this is an example with phosphorylations. You can predict which enzymes are responsible for catalyzing this modification. So if there is, for example, a specific motif that is recognized by specific kinases, there are software tools that they tell you, based on the motif that you provide, they tell you, look, based on the phosphorylation regions that you provided, these are the proteins that are most involved in putting phosphorylation in this site. And that is extremely useful, because then it's telling you, okay, I know not only that I have a series of phosphorylations that are regulated, I also know what is the enzyme that's probably responsible for it. So if I want to treat this cell, for example, I can use an inhibitor of that enzyme and see, for example, if the cell goes back to, I don't know, a wild type condition. And then, of course, actually, let me skip this because it's a long story. This one is more useful. Another important information that you can have, and now you can do it for a lot of proteins, because for a lot of proteins we have a 3D structure. For a lot of proteins we know what kind of domains they have, which domain is responsible for binding, which domain is responsible for a catalytic activity, and so on and so on. And what can be very useful is to know where your modification is into the protein. So, for example, this is a study with acetylations. This is acetylation of a protein called P300, which is actually an acetyl transferase that can get acetylated itself. And then we found regulated acetylations, right? And then what you can do, you can map these acetylations in the protein sequence, and you can check, for example, if one of them is inside the active site of the enzyme. If this acetylation is inside the active site of the enzyme, probably this is the one that is most involved in regulating the activity of that protein, right? I mean, of course, it's not exact science. It can be that some modifications, they have an important property, even though they're in a different region. But it's always helpful to prioritize your target. And then knowing that a specific acetylation is in the active site of the enzyme, it could be the most useful acetylation to start, for example, mutating or start, investigating for your next paper. So overlaying modification and protein domains, modifications and motifs, these are two exercises that they can help you narrow down the biological function of these modifications. And here is just a list of databases that you can use to map protein domains. So you can go on this website, paste your protein sequence, and then it tells you, okay, in this sequence, this is where a certain domain is, another domain is. Right. So, this is just to show you how complicated it can get. You're going to get these slides so you can, you can get some inspiration from some of your data representation if you are interested. I always wrote in the bottom here what kind of software is used to generate a graph like this. Most of this software, they have a graphic user interface, so it's enough that you paste your table with the, in the exact format, and then it makes, graphs, for you. Right, and this is just an example to tell you how, when you combine all the properties that I told you so far, can become extremely informative. So this is just a study, very briefly, where we took synaptosomes, so the end of the neurons, that's where the informations are transferred between cells. And then the synaptosome was stimulated with with the salt that is supposed to activate it, right, and activate the ion channels. So if you purify these synaptosomes and you study protein abundance and protein modification, here of course we focused on phosphorylation because they are the fastest modifications that they are transferred between between vesicles. And for example, you split your sample between proteins that are soluble in water and proteins instead that are soluble in organic solvent. The proteins that are soluble in water are proteins that are in the cytosol of this vesicle or the cytosol of the cell, while proteins that are soluble in organic, they are instead proteins that are normally associated to membranes, right? So because proteins that are associated to membranes, they're usually very hydrophobic because they're surrounded by lipids. So usually when you purify these proteins, they're not very soluble in water. So if you put all of these properties together and you check for, you group your data in, based on these different properties, you can even study, for example, which proteins they get modified by the stimulus, but they also change location, right? So these, for example, are very important proteins because of proteins that are in standby without the stimulation. When you stimulate them, they get phosphorylated, they get on the surface, on the membrane of the vesicles. These are the ones that are recognized by the other synaptosomes in the receptor end. Or the other way around, they can get dephosphorylated and then they are transferred to the membrane. And you can group the proteins based on these properties that you find. That helps you narrow down, okay, what are the proteins that are most likely to be involved during the stimulation in transferring signal. Just an example. And finally, I'm going to leave you this table. It's almost 9 a.m., so we're going to stop soon. I'm going to leave you this table where you can see which software to use to extract motifs, which software to use to extract, for example, predictions of localization of modifications, information about modifications that we already know, information, for example, about what kind of kinases are involved in X and Y, depending on the motifs that you find to be enriched. So I'm actually at the end. So if you guys can resist 5 more minutes, I wanted to spend the last moment of the presentation showing you a practical example about how you take the output of a proteomic analysis and how this output is converted into enriched protein groups. So everything we discussed today at the class, let me see first if there is any question. I see something in the chat. Okay, no problem. Okay, so the last 5 minutes, I wanted to show you something about how do you enrich proteins, according to their regulation. So this is this is an output table that you get from a proteomic search engine. So these numbers, these are the protein quantifications. Actually, this data have been already normalized and transformed. That's why there are some numbers that are negative, that's because it's after the normalization. And another thing is these data are actually in logarithm. So this is a log2 quantification. So remember, we learned this in high school and then we forget. If you want to make a ratio between one value and another, and the values are in logarithm, you need to subtract, right? Because if you remember, the logarithm of A divided by B is actually the same as doing log A minus log B, right? So this is, these values are already in logarithm. So if you want to make a ratio, you subtract. So let's say that we want to plot a volcano plot, right? And here we have two groups. So one group is this romidepsin treatment. Now it doesn't matter what this is. And this is untreated sample. So let's say we want to plot romidepsin versus wild type, right? So you can do it in Excel, you can do it with any software. I don't mind, as long as it's clear for you why we're doing it in a certain way. So let's say you take the average of your replicate in romidepsin, and then you divide, these are log values, so you subtract the average values in wild type. You get a number, and you get a number, a fold change. This is the x-axis of your volcano plot. You can sort your table by this property here. And now let's say that I want to display this. You see that, you remember this graph that you saw in the beginning of the presentation. These are the proteins that are more abundant in romidepsin. These are the proteins that are more abundant in the wild type. But now you want to display not only how much they change, but also how reproducible this observation is, right? So what you need another axis in your graph. You need a p-value. The p-value, there are, now we don't have time to talk about all the different types of statistical tests. Say the default is the t-test. It works well when you have very few replicates, for example, only 3 replicates. And then here you can pick, the first group, the second group, and then in Excel is asking you 2 more information about what kind of t-test you want to apply. Here, by default, we pick 2 tail because it's more stringent. And then here, by default, there are 3 types of t-test that you can use in Excel. Again, we don't have too much time to speak into details. When you don't know what you're doing, usually choose type 3. And then I'm going to send you a paper made by us where it specified clearly what type of t-test you should use in which situation. So you get a p-value. A p-value, of course, it's smaller if the changes are more significant between the groups, right? And then you see, for example, this one is significant because when you look at the values, you see that there is a big difference between this group and this group. So it's good that this p-value is significant. However, it becomes difficult to plot this as a volcano plot because right now, the more significant changes, the smaller is the value. So if you plot a volcano like this, where you have the x-axis that is the fold change and the y-axis that is just the p-value, you get a volcano that looks like this, where the most significant changes are here close to 0. That's why usually we don't plot the p-value, we plot the minus logarithm of the p-value. And then the base of the logarithm actually doesn't matter, it can be base 2, base 10, it depends on how stretched you want it to be. Let's say base 2. And now this one looks like a volcano plot, right? So here you have the fold change, here you have the proteins enriched in romidepsin. The higher they are, the more significantly enriched they are. If you want to know which ones are these proteins, here on the top, what I usually like to do, I like to create an additional column where I used to sort my data for proteins that have the best combination of fold change and p-value. So basically the product between the two. This number doesn't really mean anything, but it's useful. For example, if you want to sort your table for proteins that have a strong value for both. Right? For example, like this. Here I sort by this property. And now you have on the top of the table proteins that have, actually, let me do a bit of color coding. So it's helpful to know what we are talking about. So here, I colored in green the p-values that are more significant, and I color in red the strongest positive fold changes, and then at the bottom of the table, you have the strongest negative fold changes, right? So now you want to show what are my proteins up here, the ones that have the best combination of fold change and p-value. Just add data label, the Excel by default will show as a data label the value of the y-axis, right? That's not really what you want to display. Actually, what you want to display, for example, the gene names. And then like this, here is displaying, these are the proteins that have, the strongest enrichment. And then, just to reveal your secret, what this was, this romidepsin is a drug that if you take cells that have silent HIV, you can reactivate HIV expression. And in fact, you see that what we have down here, we have the HIV proteins that are being re-expressed in presence of romidepsin. So this is also sort of like a positive control. And so I will stop here. I'm gonna send you guys a paper for your reference that I don't think I sent it before. Here, this could be helpful for you for the part that we discussed here at the very end. So here, if you want to, if you have a raw proteomic table and you want to know, for example, in Excel, how you can transform it, how you can make it ready for proteomic analysis, what kind of statistical test you should use, depending on how your data are distributed. And of course, here it explains how you choose how your data are distributed. And then at the very end, there is also a power analysis, which we haven't discussed, but power analysis is that test that helps you tell how many replicates do I need to have something to be statistically different, right? So that is a question that normally you wonder, if I want to see a significant difference between this group and this group, how many replicates do I need? There are some tests to actually calculate that. I'm going to send you this paper at the end of, together with the slides at the end of this presentation. And yeah, let me stop sharing for a second. And yes, so basically I was thinking to end it here. And if you guys have some questions, you let me know. Otherwise, good luck with your, I don't know, do you have an exam or something at the end of the course? Maybe not. Oh, I know Wagner left. So Isabel, you are the host now.
Thank you, Professor, for the class. I'll open for questions. Então, pessoal, vocês estão me ouvindo?
Estamos sim, Isabel.
Então, o professor Wagner teve um imprevisto, precisou sair mais cedo, então eu vou abrir para perguntas. Alguém tem alguma pergunta? Any questions?
A question on the chat that is asking the name of the paper. So you know what? How do I do it? Let me check in my emails if I have your contact, Isabel. So I'll send it directly. To. Oh, I see. I actually got an email where there is a lot of names. I think that's you guys, right? So I'll send this paper attached to this email. So because I'm also not sure if you need a special subscription to download it, because it's a weird journal. Anyway, I will attach it here. Paper we discussed today, and you guys let me know if you don't receive it. Okay. Another question. Philip is asking, when I collect the data from the mass spectrometer and put the data in Progenesis, yes, for those of you who don't know, Progenesis is a software to align chromatograms and extract protein quantification. The proteins that are called similar to protein X, could you consider that this protein has undergone some modification? One second, I'm not sure I fully understand the question. I see. So basically, I guess what you're saying is that the protein that you find has a sequence that is similar to a protein sequence that we know, but is not exactly that sequence. And then the question is it possible that we don't identify that protein sequence, the one we expect, because there are some modifications on it? And the answer is absolutely yes. It might not be the only reason, but in some cases, as we discussed in the very first class, some proteins are really heavily modified, so much that if you don't consider some modification, you might not identify those proteins at all. Histones is an example of proteins that are so modified that if you don't consider acetylation and methylation, you might not find histone sequences in your result. Another one are surface proteins. So, for example, now it's very famous, the spike protein of the coronavirus. The spike protein of the coronavirus is on the side is fully decorated by glycosylation. If you don't consider glycosylation as a potential modification, or if you don't cut out the glycosylations from the protein, you might map a very small portion of the spike protein of the virus. And that is true for most surface proteins in any cell. Like surface proteins, they have a lot of decorations with glycans. So usually when people study these proteins, they remove the glycans using enzymes, like for example PNGAZ. But yes, it is absolutely true that a protein sequence that you cannot identify, the protein sequence could be there, but if it's modified, it doesn't show up in your list. I hope that answered the question. If not, let me know. And then another question from Dimitri: is proteomics data analysis computationally demanding? What kind of computer infrastructure are required? Okay, yeah, that's a good question too. Everything I show you is done. I do it with Microsoft Excel. I'm not good at coding. I'm very jealous of people that are good at coding. And eventually, if you write a script to process data in a certain way, it becomes faster. In the future. I usually teach to my lab members how to do it in Excel, not because I prefer Excel than R or than Python, but because I want them to see, what is going on. So why data are transformed in a certain way? How do we enrich for the proteins that have the highest enrichment, like we discussed? And then once this is clear, you can use whatever script you want. I just want to make sure that you know what the script is doing. And regarding how long it takes to do this calculation, like you saw, now doing it live, to plot a p-value, to plot a false change, it takes a second. So it's not computationally demanding. It becomes more computationally demanding, the database searching part and the extraction of the chromatograms. So that is usually something that if you do it in a normal computer, it can take several days, especially if you have a large proteomic data set. Usually we do that on a server, and you can do it in a normal computer, it's just a little bit slower. But once you have your final table, to convert your table into meaningful biology, that is, these are not computationally demanding calculations. So these are usually much quicker. Of course, we can do all kinds of things. Some things are more complicated than others, but everything we do is done with a normal computer. I see one more question here. There is an article that says in the description of this. Oh, sorry, 1 second. Let me see if someone is not muted. In the description of string protein-protein interaction, node size indicate different levels of fold chains relative to each individual network. Okay, so actually, I don't know the answer to that question. I do know this though. So when you plot a network, whatever it is, even if it's a network between what are my best friends, it doesn't need to be protein network. Usually a network is divided into properties, the nodes and the edges. So the nodes are basically the balls that we were looking at, and the edges are the connector lines that connect the balls with each other. And in string, what you have seen me showing you, in string, the only thing that changes by default is the size or the color of the edges of the connector lines if you just paste a list of proteins. I know that in string, I never tried it, but I know that in string you can also provide the protein fold change, not only the list of proteins, and then that probably affects the size of the node, the size of the bubble. I actually don't know. You probably need to try, but regardless, if you upload your network in Cytoscape, the software that I told you that you can use to customize your networks, once your data are there, you can do whatever you want. You can play with the colors, you can play with the sizes, you can play with the position in space of the of this node. If I can give you an advice from today, if you're planning to plot networks in the future, Cytoscape is the best software to get familiar with, and it's easy to use. The people have worked for many years with that. There are many apps to simplify your data representation. There you can really customize the way you like the node size. Although Roberta is right, it is possible that string does that automatically, but I have to say I never tried that. One more question. Thank you, guys. Do you know if there is any software? Do you know if there is any software that are able to model and predict protein-protein interaction based on your peptide sequence? Or this analysis? That's a good question. That's probably a question more for like a hardcore biochemist. So Bruno is asking if there is any software that if you know the protein sequence, if you can predict whether these proteins form interactions with each other. Of course, there are software that I can send you some links that if you paste a protein sequence, it predicts the structure of them. Whether it predicts if these proteins form an interaction, I don't know any that can do that. It can be just my lack of knowledge, but that sounds to me something that is very prone to very large errors. So that's probably why there are no famous ones around, because probably the probability of creating false positives is very high. I might be wrong, but I honestly think to the best of my knowledge, I think they don't exist. If they exist, please let me know. I see there are some raised hands. Reynaldo and Isabel, you guys want to unmute and tell me? Okay, in the meantime, let me close 1 second the door of the office because there is some chatting from outside. Okay. Yeah.
I'll go first. So since we are talking about application, biological application and clinical application, looking just at the proteome might give us a picture very incomplete. So what do you think would be the very best approach or tool to analyze more than one omic data?
Yeah, multi-omic analysis is something that most, a lot of scientists have thought about it. So now we have this big, transcriptomic data. We can do metabolomics, we can do proteomics. Like, can we integrate them together? That's a very good question. And people have tried. The real problem with these comparisons is that people have not done something that is much more than just correlating data. So when you make a correlation, in the end, correlation analysis is not very informative because it only tells you if two data points go with the same trend or go with different trends. that can be informative, but it's also true that, you know, some proteins affect some metabolites, but they don't necessarily change their relative abundance. So integrating multi-omics is the way to go, definitely, but it's still very challenging. And in fact, let me tell you this, you know, informally. I think this is the challenge that you guys can embark on, you know, for the years to come, you know, to try to conjugate and make sense of multi-omics data sets, because honestly, nobody does it right today. I have a friend of mine that they work in pharma companies. They're very happy there. They work at Roche, they work at Merck, they work at Johnson & Johnson, and they tell me all the same thing. They tell me, we have databases here that are full of patient data of all sorts, you know, blood metabolite, pressure, you know, phenotypes, all kinds. And we have the problem that we don't know how to deconvolute this data into a biological meaning. Like, we have all of this data, they occupy databases that are bigger than the database of YouTube. But the problem is that how do we conjugate, for example, DNA sequencing with blood pressure, right? And they have some sort of properties that bind them together, but we cannot find the proper transformation of the data. And that's something that, you know, I actually think that the people of your generation will find the solution to that. Because these data, even just proteomics and metabolomics, they are hard to correlate, because it's not necessarily true that one protein regulates one metabolite. There is so much going on, and you know, in order to connect them, you need to know more about the data you are looking at. But yeah, I agree with you, Isabel. I think proteomics is only a partial view of the, you know, of the phenotype of the cell. It's still extremely useful for a lot of studies, right? So if you know which proteins are regulated, you have some targets that you can prioritize, for example, for drug treatment. And you know, and that's how we have done until now, and we had, you know, a lot of good results as well, you know, in science. But you're right that in order to have the complete picture, you need to integrate this multiple big data. But there is not a simple way of doing that. And to be honest, my perspective is that nobody's doing it right yet.
Thank you.
Reynaldo wanted to ask a question.
So, hi, Professor. Thank you again for the lecture.
Hello.
I have some questions. I will start with some connection. So, I see some people using nowadays more the term of multiomics, but sometime in the past and nowadays also I see some researchers use the term systems biology. I wanted a crude question. What is the main difference about these two terms?
Yeah, so I think I think system biology, but again, this is my personal opinion. So please, you know, make up your own mind as well. I think system biology is a term that is a little bit abused, in the sense that system biology, by definition, means how a biological individual behaves in a system. So this basically means that, for example, if you're studying a cell, you're not only studying what's going on inside the cell, you also need to know how the cell behaves in relation to what is surrounding it. If you take, for example, an individual, like if you do systems biology of me, for example, it's not only how healthy I am and, you know, what is my blood pressure, but also how I am behaving in relationship to where I am right now. You know, what are the people I'm talking to? What is the weather in New York? This is systems biology. So is the biology of something inside a system. So inside the multi-layer complex, environment. And I understand why people use the term systems biology sometimes to define, because we're not looking only at one group of molecules, but we look at multiple types of group of molecules. But the definition of system biology is what I told you. So I feel like many times it's been a little bit abused. When you use multiple data sets, you are not really doing, for example, if you're using proteomics, metabolomics, but you're looking at all of them with the same cell, you're not really doing system biology. What you're doing, like you say, you're doing multi-omic of a biological system, but not in a system. So I don't know, maybe I got confused, but that's what I meant by the difference. I hope it's clear, right? So
no, I understood. So in the same matter, do you think, for example, that there are some softwares that try to merge clinical data with omics data or other measurement types? They try to normalize it and merge it. Will be something a little more near in systems biology term.
Yeah, good question.
Not systems biology, but a little more near. Do you think?
Yeah, I don't know how I would call that. I know of some software that if you have regulated proteins or regulated RNAs, you can put them on those software and they tell you whether your regulations are similar to a certain disease. One is we briefly mentioned is this GSPA. I wouldn't call that systems biology. Although to be honest, you're not going to jail if you call it that. People can call whatever they want. But I would say that rather than systems biology, that is an example of translational application of your data. That's how I would call it. More than systems biology. It's the same for machine learning, right? So some of the graphs that I show you, for example, the graph where I show you that there were this group of proteins that all have the same trend, that is a sort of machine learning. But machine learning is also a term that is used a lot, and it's used a lot because it sounds cool, but it includes a lot of different tests that most of them are basically clustering. And unsupervised clustering, they have very specific algorithms. For example, k-means is an algorithm that does unsupervised clustering. And calling that machine learning, I think, is just a way to be vague about something that is in reality very specific. So when you use the term systems biology, I'm not saying it's wrong, but I'm saying that I think you convey your message more accurately if you actually name it what you're actually doing. Because these terms are very generic, very cool, but they don't inform you enough about what you're actually doing.
Yes. Okay, thanks, Professor. I guess I started with a philosophical question. I will retain in more technical matters now. So let me see. Oh, okay, another philosophical one, I guess. Sorry. You talked during the lecture about fold changes and p-values. What do you think about fold change cutoffs that are sometimes required for papers published?
Yeah. No, this is a good question because it's a question we get very often. Okay, so the idea is, okay, when I have a p-value, what is a good p-value? Or when I have a fold change, what is a good fold change? And the thing is, there are some general, not rules, but let's say there are some general agreements of people. People say, okay, a good p-value is a p-value that is smaller than 5%. smaller than 0.05. That is the threshold that everybody knows, and we usually use that without really knowing what it means. But let's say it's fine. And regarding fold change, it's even more arbitrary. People say, okay, 2 fold change is good enough. So, but this means that 1.8 is bad and 2.1 is great, right? I find that this approach sometimes necessary to simplify your data, to not get too much into the weeds. But if you think about the class I gave you today, the way we organize our data, you actually don't need to choose a very stringent threshold. In the end, when we sorted our table by score, this means that we sorted our table in a way that on the top you have proteins that have the best enrichment, and on the bottom proteins that have the opposite enrichment. Now, your table organized like this is already useful to perform functional enrichment. You remember Gorilla, the software? You can upload the rank list of genes, and then it's telling you what are the functions of the proteins at the top of your table. If you do it like this, you don't need to chop your table and say, okay, this is good, this is bad. But even your sorting becomes quantitative. So in this way, basically, you don't need to penalize proteins that for 0.1 value they don't pass the threshold. You just organize your table in a proper way. So bottom line, I think that sorting is a much stronger way of efficiently grouping data compared than filtering. I would do sorting rather than filtering. Then, of course, you need to get a feeling, like if you see that all of your proteins, they have a p-value that is very bad, then, you can sort this data, but keep in mind that probably the result is not very meaningful. But if you have significantly regulated proteins, I would sort them rather than cutting the Volcano plot. I think it's more useful this way.
Yes. Thank you, Professor. That's really clarifying. I will, if you allow me, may I ask just one more question?
Yeah, let me just check. I also want to make sure that everybody has. Okay. If you guys can wait just one more second, Reynaldo has one more question. Yeah, go ahead, Reynaldo.
Sorry, guys. So regarding these new instruments that acquire massive amounts of data about mass spectrometry, recently I get some data that come from a fusion, I guess. This data was in centroided data in format. This was not profiled. And I would like to know if this is usual to reduce the data size in this kind of instruments, or this was a peculiar case.
You say that the spectra were centroid and not profiled. Is that what you said?
Yes.
Oh yeah. I mean that is a very technical question. So the difference between saving your spectra as profile or centroid, it basically means how many data points per spectrum you want to save, right? When your molecules they hit the detector, they hit them, with a certain resolution. When you run with any analyzer, you can pack ions of the same type into more or less compact distribution of ions, right? So in the end, if you want to see the resolution of your analyzer, you need to plot all the data points that define the Gaussian of where your data are concentrated, right? So these are profile data. So you see not only the mass of your molecule, but you see also how much it deviates from the central value. And this saving spectra in profile mode, you save all of these data points, but obviously these spectra are much heavier in terms of file size, right? Because for each signal, instead of having one data point, you have maybe 50. So that's why if you save spectra in profile mode, the files they become so big in terms of gigabytes. So that's why most people now save data in Centroid, because Centroid does not save the profile of the peak, only saves the central value. And then instead of having 50 data points, you have only one. And this is something that people do to make files smaller. If you need to know the resolution of your spectrum, then you need to save your data in profile. But to be honest, when you perform database search, the software doesn't care about the profile of your peak. So the reason because people save data in Centroid is because it's just easier for the downstream proteomic analysis. So yeah, we do that too. We save our data in Centroid because we don't need to know the profile of the peak. When we need to know, of course, we save data in profile.
Okay, thank you, Professor.
Thank you.
There's in the chat.
Yes, so there's one more question in the chat from Carolina. The IGPS software uses only the phosphorylation motifs that generate the score. Yes, absolutely. So this is a very good question. So the thing about this, I show you an example of software that predicts which enzyme is responsible for putting a modification depending on the motif of that protein sequence. One of them is IGPS for kinases and phosphorylation, and it's absolutely true that the same motif sometimes can be identified by different kinases, right? So you don't have like exact matches between a motif and which enzyme is responsible to modify that motif. So it's absolutely true that there can be multiple kinases that potentially modify that site. Regarding which one is the most probable, these software, they don't tell you which one is the most probable. They usually tell you, actually no, let me rephrase. These software, they give you a score for each match, and then each score is supposed to tell you which one was the kinase that most likely modifies that site. So you can use that score, but keep in mind that that score is drugged, is biased a little bit, depending on how much we know about each single kinase. What I would do instead, because when you do a phosphoproteomic experiment, usually you have hundreds, if not thousands, of regulated phosphorylation. Rather than using those single scores, I would check how many phosphosites are regulated by the same kinase. Because it's very unlikely that one kinase modifies one protein. Usually a kinase modifies a lot of proteins. So if you have a lot of motifs in a lot of different proteins, and there is one kinase that is constantly repeated, that is modifying this protein, this protein, this protein, this protein, I think that is more informative than just the differences in score for two kinases on one specific site. So I would use which ones are the most frequent. Okay, I have one. Okay. Okay, so that's a different question. Okay, yeah, so that's my answer to the prediction software. And then Carolina has one more question that says about label 3. We have a limitation on the number of conditions we can run in one statistical analysis because the test loses strength with more than 4 or 5 conditions. Do you think it would be feasible to do 2 different statistical analyses divided by conditions? Yes, absolutely. So that's another good question. Unfortunately, in one class, we don't have time to go through all the possible statistical tests, but it is true. So when you have multiple conditions, and all of these conditions are independent from each other. For example, you have drug 1, drug 2, drug 3, drug 4, etc. So you don't have a time series, and the samples are not connected by some properties. When you're doing the analysis of variance, the analysis of variance gives you one p-value, and that p-value usually means one thing. If you do the one-way ANOVA, for example, that p-value tells you among the 6 conditions that you are comparing, there is at least one that is different from another. That's what that p-value means. So this means that if you have 20 conditions and you have a significant p-value, it can easily be that you have 18 samples that don't change at all, and then there are 2 that change. And that p-value does not tell you which one changes. So it's true. When you have a lot of conditions, you have to be careful when you do the analysis of variance, because it will be eventually significant, but the significant p-value has less and less meaning if you are comparing a lot of things together. Then I would think more carefully about what you really want to know in terms of what is different from what. For example, if you have your drug number 5 that you think is the most effective, then you can do a 2-condition comparison where you have like the drug number 5 versus all the other conditions, like the drug number 1, 2, 3, 4, etc. And if you have a significant p-value, then it tells you that there is, that protein, whichever protein you're looking at, is significantly less or more abundant in drug 5 compared to all the others. Now, when you have all of these different conditions in the same pool, very likely these data are not going to be any more normally distributed, because you're going to have, this is not an homogeneous population, right? It's a population of multiple conditions. So when you reach that level that you're comparing one group with multiple groups, then you probably need to switch to non-parametric statistics. The equivalent of the t-test for a comparison between 2 groups is you can either use the Wilcoxon test or the Mann-Whitney test. In the paper that I sent you, there is an example. So you can see, when to use the non-parametric test when you're comparing 2 groups. But yes, bottom line, yes. Yeah, bottom line, I agree with Carolina. When you have a lot of conditions, you have to be careful in, in what kind of statistical test you do, because even if you get a significant p-value, that p-value might not have the meaning that you want it to have. I hope that answered the question.
Thank you, Professor.
Thank you, Carolina.
Hi, Professor.
Hello.
Great presentation, thank you. I have a question about a problem that I usually have with my protein lists in interaction softwares. Usually like about 30 to 40% of my list is found to be missing in the interaction software. So I don't understand really why this happened, if it's because it doesn't have a lot of data about it, and what percentage is considered to be good enough, if I should choose another approach, or if there's something that I could do so the software could find like easily my list.
Is that a proteomic study or
yeah.
by modification?
Yeah, and usually like in string software.
So missing data, you mean that you have empty cells in your quantification? Is that what you mean by missing data?
No, like I put my list of proteins in there, and they're like for 30, it only finds like 10. And the rest of them, I don't know what happens.
Or you put it in string, you're saying? But
yeah.
Okay. Is it a strange organism?
It's human, usually.
Oh.
Fine.
Yeah, that's strange because.
Yeah, usually, and it's sometimes I have like very known proteins, so why I couldn't find it? Because I think both of them, the lists come from UniProt database. So if I could identify why the software doesn't find.
Yeah, that looks more like a technical problem. So what happens sometimes, what you can do, you can send me that list, we can look at it. But usually what happens sometimes is that some of these online tools, they don't recognize your input because for some reason there is some strange character that they are not used to. Maybe you have isoforms. Actually, I don't remember if string recognizes, if you have like the accession number -2, -3. I don't know if string likes that. Usually I remove all of these strange characters because anyway, there's not going to be, string is not going to find different properties if you have a protein with isoform 1 or isoform 2, because for string is the same protein. So it's actually useless to specify the isoform. But exactly what is your problem, that I don't know. But if you send me a table, we can look at it. But usually the problem, if it doesn't recognize so many accession numbers, usually it's because there is something wrong with the format of the input file, because string should have all the human proteins. It's strange that 40% of human protein are not recognized. So it's probably the input format, I would say.
Yeah. Okay.
Yeah, send me the table. I'll be happy to take a look.
Yeah.
And
I will. Thank you.
I reply to everyone, so that's another piece of the class. If we find the problem.
So I think that's it. I think there's no more questions. I want to thank you. I'm sure the last few classes were very enriching for everybody. For me, certainly was. Thank you. I wish you a great semester and be safe.
Thank you, guys. It was a pleasure meeting you. So have fun with the rest of your semester. See you soon. Bye.
Bye-bye.